our group decided to look at the paper titled "The Elongation Factor EloF1 is Required for Mammalian Gastrulation." This study was done in the Department of Veterinary and Animal Science at the University of Massachusetts Amherst. I'm Leanna.
I'm Erin.
I'm Isaac.
Okay, so our given topic was regarding gene structure and function. Specifically, it's important to understand how gene structure immediately impacts gene functionality, thereby expression. Attempting to analyze unknown genes opens the door to solving medical mysteries, understanding mechanistic phenomenons, as well as helps with developing new forms of treatments. And of course, the list goes on and on. This paper represents no better example, as its main research goal was aimed at answering the question listed here. What is the functional significance of the loss of function allele for EloF1? EloF1 is a mammalian gene encoding a subclass of transcription factors interacting with RNA polymerase. And this interaction helps to lengthen the transcribing RNA strand. These guys are also known as elongation factors. The term EloF1 stands for elongation factor homolog 1. This is important to understand, as studies have already been done surrounding ELF1, which is why EloF1 is considered the homolog. ELF1 has been found to play an important role in histone displacement, preventing proper RNA polymerase activity, and alternatively splicing isoforms. Because analyses have already been done regarding the yeast homolog to the gene of interest, authors of this paper hypothesized that ELF1 would be regulating alternative splicing in mammal cells as well. Because this hypothesis is stated at the beginning of the paper, they identified EloF1 as having expression similar to that of ELF1. When looking at the experimental design for this paper, the scientists sought to analyze mammalian expression of EloF1, as well as the morphological and molecular characterization of the homogeneous EloF1 knockout embryos in early development. The researchers chose to study gene functionality using the mouse as a classic model for identifying in vivo gene function. But before continuing to other parts of this presentation, understanding gastrulation is essential. Gastrulation leads to the formation of the 3 germ layers. These germ layers are known as the ectoderm, mesoderm, and endoderm, which help further development of different organs and organ systems within the animal body. Identifying how genes and different genotypes of EloF1 impact gastrulation will give reasoning behind functionality. To do this, they created and genotyped their mice embryos, and it was from these embryos that they were able to extract RNA and therefore use it as a template to perform reverse transcription PCR, turning their RNA into DNA for PCR analysis of target genes. Then, using histological examination, immunofluorescence, imaging, and staining, they were able to comparatively examine knockout alleles of EloF1 in expression and embryo development. With this in mind, our authors set up an experimental design not to study the gene specifically, but to study the gene's expression by creating knockout alleles that can elucidate function.
So one of the first major findings was that EloF1 knockout embryos show severe morphological defects. So what figure A is showing is that researchers replaced portions of exons 1 and 2 with a LAC-Z reporter to create a mutant EloF1 allele. Later in the presentation, I'll get into why and how the LAC-Z acts as a reporter. But essentially, what the big takeaway from this is that heterozygous crosses fail to produce any homozygous pups or embryos, which demonstrates that this genotype is lethal in early embryonic development. This is also shown in figures B and C, where crossed EloF1 heterozygous animals and dissected litters at embryonic days 7.5 and 8.5, that's what the E stands for, is embryonic days, show very significant morphological defects. Researchers also wanted to ensure that EloF1 transcription was altered as expected, and so they performed RT-PCR, or reverse transcriptase PCR, and used intron-spanning primers to amplify the wild type transcript. And so beta-actin was just their housekeeper gene, but what figure D is showing is that it confirmed that all embryos with an abnormal phenotype also lacked an endogenous EloF1 transcript. They also found that EloF1 is differentially expressed in embryonic and adult tissues. So figures A, B, and C are whole mount analyses at embryonic days 6.5, 7.5, and 8.5, and these are And then from these whole mounts, they took sagittal sections and stained them for XGAL. So they were able to do this because of the LAC-Z reporter gene. So XGAL is going to be shown in blue, which is shown in A prime, B prime, and C prime. And the idea is that because the LAC-Z gene was within exons 1 and 2 of the EloF1 gene, anytime EloF1 is expressed, we will see that blue color. And so at day 6.5, which is shown in figure A prime, LAC-Z expression is strongest in the epiblast. At day 7.5, the reporter is expressed in all epiblast derivatives, including the embryonic mesoderm, neuroectoderm, and definitive endoderm. And then at C prime, the majority of the tissue showed no to weak LAC-Z expression. Researchers then performed RT-PCR and LAC-Z staining on a wide variety of adult tissues collected from heterozygous animals to confirm that the LAC-Z reporter does in fact correlate with endogenous EloF1 expression. And so what this is showing, and again, beta-actin was their housekeeper, is that the expression of EloF1 is different in different tissues. And that's just shown by this gel right here. The negative represents their non-template control. They also found that EloF1 mutant embryos failed to initiate gastrulation. And so what they wanted to do is explore the structural and molecular defects that occur in the absence of EloF1. And so this was revealed through immunofluorescence and H&E staining. And what this revealed was that embryonic compartments of mutant embryos are filled with small and abnormally rounded cells. So they're not cuboidal or mesenchymal in shape, which would be expected. And that's observed in the control with A prime and A double prime. And so this suggests that EloF1 therefore has a role in proper embryo development, at least at the morphological level. So normal morphogenic events that occur during gastrulation are accompanied by lineage-specific gene expression. So OCT4, which is stained in green, is expressed in the inner cell mass. Brachyury, or just T, which is also in green, is a mesodermal marker. And so what they wanted to do is, in assessing the mutant embryos, which is in figure B, they used immunofluorescence to examine the expression of OCT4, T, an epithelial marker, ECAD, and PH3, which is a proliferation marker. And so what's being shown in the mutant embryos is that there's no obvious OCT4 positive cells. There are a few PH3 mutants, and then T is absent in mutants, which underscores that the primitive streak failed to initiate. And so taken together, this confirms that their mutants failed to initiate gastrulation. EloF1 also regulates alternatively spliced mRNA variants. So previous evidence shows that EloF1 interacts with other proteins to facilitate the trimethylation of histone 3 lysine 36. And so modifications are linked to alternative splicing through the recruitment of a protein called polypyrimidine track binding protein, or PTB. So PTB is an RNA splicing factor that binds to and in turn represses the use of specific exons, including a splice variant fibroblast growth factor receptor 2, or FGFR2. And FGFR2 is known to play a role in gastrulation. And so researchers wanted to investigate that altered splice variants may play a role in the defects observed in EloF1 mutants. And so in EloF1 mutants, they found a significant decrease in FGFR2 splice variants, which requires PT-dependent trimethylation of histone 3 lysine 36. And so no relative reduction in the splice isoform that is not PTB dependent was shown. And then that shows that EloF1 is involved in the regulation of tissue-specific exon choice during mammalian embryogenesis. So this warrants further genome-wide exploration.
The abnormal morphology through H&E staining and protein marker localization in the embryonic tissue sections show failure of a gastrulation event in the EloF1 mutant relative to the wild-type mouse. The expression of the LAC-Z depicts the presence of EloF1 in several embryonic layers through the stages of gastrulation. Collectively, these results support an essential role for EloF1 in the context of embryo viability. The RT-PCR experiment was designed to investigate how the loss of EloF1 may influence the regulation of alternatively spliced mRNA variants. They found an overall decrease in the FGFR2 splice variants in EloF1 and a statistically significant decrease in the FGFR2 3 beta transcript in the EloF1 mutant relative to the wild type. This, however, only answers part of the question of their hypothesis. In the quotation above, they say, "We hypothesize that the loss of EloF1 function could prevent SET2 recruitment, resulting in altered histone 3 lysine 36 methylation, which has been shown to correlate with alternative splicing patterns of genes pertinent to development." In yeast, ELF1 works with SPT4 to promote histone displacement through trimethylation of histone 3 lysine 36, allowing for the DNA binding of RNA polymerase II complex. To generate evidence for yeast ELF1 as an ortholog of mammalian ELOF1, the researchers could have assessed the presence of the mammalian SPT2 protein, a homolog of yeast SPT4 in whole embryo protein lysates. In addition, they could have used an ELISA to compare the relative abundance of trimethylated histone 3 lysine 36 in the wild type and the ELOF1 mutants. The resulting data may be taken together and used to provide support for the similarities in yeast ELF1 and mammalian ELOF1 gene structure and function. Thank you.
All right. Thank you, guys.
Okay.